question, right? And I didn't know how to modularize it to make it effective agenda item. And, and to some extent, you guys are maybe mentioning in your agenda, but at the end of the story, you remember during that meeting, our NIH officer was kind of pushing us to get a tool, right, that we can link to you guys that would kind of do stuff, something for links. So, I mean, as we go down, down this path, I think at some point it would be really good if we could somehow leverage your resources one way or another, right, for our purposes in terms of linking it to our portal, or maybe, I don't know how you guys gonna deal with, say, your super network, master network. Is this gonna be a resource that somebody else will be able to run their algorithm on and that kind of stuff, right? Yeah.
So, Mario, this is a great point. Let's actually begin with that. So the knowing analysis framework will be a portal whose backend is on the Amazon cloud. And that backend includes the massive network of prior information and annotations. And the portal will have an interface whereby you can do the sophisticated analysis, which would normally take very long on a regular machine, but because it's a cloud backend, it can be parallelized and made faster that way. Now, the demo that we did in the November meeting was of one such pipeline that was for clustering. And however, the problem was that the pipeline was not really accessible to outsiders at that point. And our current highest priority is, and that's what we're working on now, to make the cloud-based implementation usable by people other than us. If it was a simple web server, it wouldn't be very difficult. And if I'm able to use my web server, then you'd be able to use a web server as well. But it's more than a web server, and so there are some technical issues that are being dealt with, and we are working very much on that exact point. And we are really hoping to have our first pipeline ready for use. And it will do clustering in various ways, knowledge network guided clustering, so not the usual clustering. And it will, and we're hoping that you will be, someone in your team will be our friendly users in the beginning and give us feedback on that. And we really hope to have this done by June of this year. And after that, we will, once by then, since we would have figured out the technical obstacles and solutions for the skeleton for one pipeline, it will be easier for us to make the various other functionalities that we are building more specifically for links data to also be accessible to you through the. So you'll go to a web URL, not really, which will not look different from any other website, except that the backend is hosted on the Amazon cloud. And we'll figure out what to do. I think in the beginning we'll figure out hacks for the payment issues as well. In the long run, the idea is that this could be part of Commons, or NIH would have part of ways to allow the users to pay Amazon, for the cloud usage. But in the short term, I think we'll figure out a suitable hack for that aspect. Does that sound reasonable? So around June is when we are shooting for the first working pipeline accessible from outside.
That sounds very good. That sounds really, really good. Yeah.
So Could you please maybe send us in the meantime some links or some demos or whatever you think in terms of functionality that will be available, so we can start planning on our side as well, even if at this point we cannot use it, but so we're more aware of what's coming.
Absolutely. We have various levels of description of the pipeline. So, we will share that, a high level and medium level description with you. So, and also explain it to you in one of our meetings.
Okay, great. Yeah.
Okay. So if I may then proceed to some of the questions we've been having.
Charles, what was the first item? Progress and clustering of links experiments based on
geo-enrichment patterns. So this is a project that Jinfeng Zhao, who's raising his hand now, he's a graduate student. He's been working on this project, and he has a few slides that I'll use share screen to. Let's see. Oh, you might. I don't. Oh, do you guys have a wall for this?
No, we see the screen.
Okay, great. Yeah. So the idea here was to look at each links experiment in terms of the enrichments, the geoterm enrichments that of its genes, kind of like enricher does, except that the experiment is then mapped into this new space for representing the experiment. Then the new space is which geoterms are enriched in this experiment. And then in this new representation, we can start asking questions about which experiments are similar to each other, how do different links experiments cluster, and so on. So again, on the top left, can you see the cursor as well?
Right.
Right. So he took actually, just to get started, one particular cell line worth of experiments. There were 633 experiments and all genes, and the rows are experiments, and these were I think the level 4 Z-scores. These 633 experiments represented 350 perturbagens and DMSO. And then he subjects each experiment, each row, to a hypergeometry test with a 750 geoterms. So he now has a new representation of the same of the above matrix, which is experiments by geoterms. And for now, this is just a binary matrix indicating whether the enrichment is at a certain nominal p-value. So a 1 would indicate that that experiment is enriched. The genes differentially expressed in that experiment are enriched in a particular geoterm. And again, this is quite similar to what enricher and I think enricher allows you to do. So this is what was not our main goal. The goal was then to cluster the experiments and see how the different perturbagens group or segregate. So he subjected this to a simple hierarchical clustering for now. These are all very basic first steps. And then on the dendrogram, he started examining certain properties of the clustering. So what is meant by that? You would expect that the same perturbagens, if there are multiple experiments, they will tend to be in the same cluster. We had discussed this previously, that what we really expect is that replicates should cluster together, should have the highest similarity. For now, let's take some license and expect that if the experiment involves the same perturbagen, then they should tend to be on the same side of this highest level clustering. So he's looking, this dotted line here indicates the top level cluster into clustering into two groups. And so he started asking if this property really holds. That is, does the same perturbagen induce the same cluster? And the way he did that was by counting the number of edges. So what's an edge? An edge is drawn between two experiments if they are the same perturbagen. Correct, Jinpeng? And so in that sense, the edges really shouldn't be colored differently. They're all the same meaning. The nodes are colored differently to indicate different perturbagens. And so you're seeing that there's two experiments in yellow that were the same perturbagen, two other experiments in green that were the same perturbagen, and 4 experiments in blue that were the same perturbagen. And you'd hope that there are few cross edges crossing clusters. Am I making sense so far?
Yeah.
Yeah.
Okay. So then, you can you can count these number of cross cluster edges and ask how does that number relate to the expected number if the clustering was random. And the first result was very strange. The number, so this is the first result, the number of cross edges in this case were 123, whereas which is actually somewhat more than the expectation. And that was very unexpected. You would, as I said, we would have, this was the first time we were playing with this kind of an idea, but we had expected this to be less than expected, significantly less than expected, given that we expect the same perturbagen to fall in the same side of the clustering. Then he started looking more carefully, and so by the way, these other numbers are the analytical expectation, and then based on simulations, the mean. So he did some simulations to estimate empirically the mean and standard deviation, and based on shuffling of random graph for random clustering, random clustering of the same graph. So when he looked more closely, he found that the one kind of perturbagen that was really not falling on the same side was DMSO, which, as you know, is not a perturbagen; is the control treatment. And there were many. Basically, the DMSO experiments were falling all over the place in both clusters, and were messing with our nice little story. So then, we're going to ask you, what exactly these DMSO experiments, how they are done. But he took the DMSO experiments out. There were how many? Like 18 experiments, right? We took them out and repeated the exercise. And now, the number of cross edges was 51, which is, I think, about 3 standard deviations or more below the expectation, as we had initially expected. And, so that's what's shown in this table and in this histogram. Now, then he further went back to this DMSO puzzle as to where exactly was these experiments falling, and this is a PCA plot of the same vectors, and you can see that the clusters are, as expected, in separate regions of this two-dimensional plot. But the black points are the DMSO experiments. They're falling, really spread out in both regions as well as in between. So I guess the questions are, which have forced us to ask more carefully, what does the Z-score really represent that we use in any level 4 vector, and how did the DMSO control experiments fall into this Z-score calculation, if at all, and why might DMSO experiments, when thought of, when you look at the high Z-scores in those experiments, what does it really mean? What does a gene highly differentially expressed in a DMSO experiment reflect? So, any ideas on those?
So, can I ask you just a couple of questions before we proceed with DMSO start? So, how did you choose your experiments?
He took a particular cell line, which was not too many, not too few. He wanted to do some, like some cell lines will obviously have orders of magnitude more experiments. He wanted a smaller data set to begin with, and not too small either. So he took this SNU C5 cell line and took all experiments there.
Okay, so I mean, there are people better qualified to talk about DMSO in this group, but I can tell you. So what happens, I think, is that DMSO is, it's treatment that is given to each of these cells or vials or dishes in addition to the additional treatment, right? And depending how you normalize it out, right, it's really kind of, it's not a treatment on its own, right, because it's present in every other treatment.
Oh, it is? Okay.
So DMSO is what they call vehicle, right? Solvent, which is basically the medium in which you add whichever other chemical you're using to treat.
Right, okay.
So I think what you got actually makes perfect sense, right? That this DMSO, in principle, they say, yes, it has somewhat of a reproducible signature, but it's very weak. And you got it to be distributed fairly randomly, which is good, right? Okay. And in principle, I think you should drop it out and not use it. The only way I would use DMSO is that maybe if you want to create signature that is, say, comparison to DMSO, right? So compare each signature to its DMSO that were on the same plane, for example, treatment. You could think of this as an additional normalization step.
Okay.
But I would not use them in the general kind of clustering setup.
The way I understand what you said is that I think the key point which we realize is that every experiment, every perturbation experiment is already carrying the DMSO is used as the carrier for the treatment. It's a vehicle or the conduit for it's the solvent really.
Right.
And the DMSO experiment is where you just treated the cell with the solvent. It wasn't carrying any perturbagen. And it caused some changes. It did something to the cell, and so it caused some changes. Those changes are caused by just its property as a solvent. Right? So then,
Hey, Saurabh, we can hear you well. I don't know for some. Are we meant to hear you, or you're talking to your?
No, you were meant. So, but I had turned my head. So, can you hear me better now?
Yes, yes, very good.
So I guess the mistake we were making is that we were thinking that the differential expression of a gene is differential expression between treatment with perturbagen and treatment with DMSO, and that the perturbagen did not involve DMSO.
Right. So you can think of it. This is actually in the newest releases of this data. Level 4 data is defined in two different ways. One way is the way this data is created, which was their previous period data, where the data is normalized to the population mean, right, of all experiments on a single plate. In the other version, this data is actually created by comparing the profiles to DMSO profiles, right? So nowadays, you can do it both ways, basically. So for example, when Avi creates his signatures, these whatever his method is, the characteristic direction signature, he actually compares treatments to DMSO to create a signature. But in the general, pardon?
How? So I mean the DMS. So he'll compare it to all DMSO experiments, of which there are several. We saw in this cell line there were 18, and we don't even. So you're saying that they're all very weak, weak signals, and so comparison would be to a near null signal.
Well, it's a very weak signature, meaning that these scores are relatively small. Like distribution of these scores for DMSO should be very narrow.
Okay.
In comparison to the distribution of other treatments. That is something that Broad is saying is the case. I actually never looked at it, to be perfectly honest.
Okay.
But they are saying, you shouldn't really, DMSO has somewhat of a reproducible signal, but it's very weak, and you just don't treat it as a perturbagen, right? Because it's kind of incorporated in the signal of all other perturbagens, and you just don't analyze it in the same way that you would analyze others.
Okay, so then one other point I wanted clarification is the Z-score you said is a normalization of that gene's expression relative to the whole population of what? What was the population?
Right, so here is how the Z-score gets created. You have a 384 plate, right? In each plate is, so one cell line per plate, so it's the same cell line.
Okay.
And so probably the data that you used was generated by two plates, right? It's a replicate of the same plate, most likely, but I'm not quite sure. So in each of these wells, you put one perturbagen, or maybe in
DMSO.
And DMSO, right? So whatever. So each profile at the end comes from one of these wells on a single plate, right? And the way you normalize these now, so you measure, right, the intensity, fluorescence intensities in each well for each gene, and then you perform do the quantile normalization of the profiles that you get, right, for these 300. So you have 384 profiles that you measure on a single plate.
Yeah.
You run quantile normalization on these profiles, and then you calculate per gene these statistics for these 384 measurements for each gene. So basically, for each gene, you subtract the median and divide by
The mean.
No, they actually subtract the median. It's a robust estimate. So they call it Z-MAD. Actually, it's a robust Z-score. It's you subtract the median and divide by robust estimates of the standard deviation.
Okay.
But it's done gene by gene, right? So that's why it's populationally normalized, right? Because it's kind of
the meaning of the Z-score then depends on which perturbagens went into that plate.
Yes.
Okay. Okay.
Yes, but the idea is that, again, the idea is that the population is sufficiently big.
Right. Yeah, I didn't know that. Okay.
So these scores of differential expression, maybe receive it, but as over or under from the.
Right, relative expression. Yeah. It's not like when we think of differential expression, we always think of with treatment, without treatment. You know, this condition without condition. But that's not what the Z-scores represent.
That is correct. I mean, you can construct if you want to. Again, and some people think that that's the way to go. Rod originally thought that this is better, and I think they still believe that the populational normalization is better. But if you want to, you can say the way that you could do it is because there are multiple DMSO measurements on each plate, right? So you could kind of run statistical combination by comparing first within the plate, right? Treatment versus DMSO, and then combine plate replicates, right? And to come up with a statistical kind of estimate of, or a more, I don't know, the treatment versus control kind of measurement.
It's we don't use the DMSO we don't think of it as a perturbagen, which is with an expectation of clustering together.
Oh, why the DMSO? They are.
We can't hear anything.
Yeah, I'll repeat it. Go ahead. What's the question?
If you put the same stuff into 18 different experiments, then you should expect that there are different. Well, but it will depend on how the experiments work, because it's populational normalization.
Right. No, but that is, so he's still curious why these 18 experiments end up giving different experiments. And I think, maybe the enrichments are weaker, and you're playing on the random scale. So things are just different because they're random.
But if the signals are weak, because at the beginning we dropped all the Z-scores. I mean, when we start with the matrix of experiments versus human genes, we dropped all the Z-scores below 2.5, right?
Right.
So everything.
But still, out of 20,000 genes, maybe there's a chance that 50 of them are going to have a Z-score greater or less than 2.5.
Yeah, I think it's okay. We can discuss this later.
Well, but if I may add something, because the distribution of Z-scores and what happens with DMSO is not going to be only dependent on what perturbations you have on a given plate, but also their concentration.
Right.
Depending on whether you use higher versus low concentrations, you may also see the effect of DMSO seemingly less random versus maybe more random, because you're actually diluting these chemical moieties in DMSO with different concentrations. Sometimes it could be very low concentrations, mostly around 1 micromolar, but sometimes can be low, sometimes can be much higher concentration.
Okay.
And DMSO itself is a chemical perturbagen of sorts. It's a weak perturbagen, but it does perturb things.
Right.
And the scale of that perturbation is going to depend on also on the concentration of these chemical moieties that you're diluting in DMSO, right? Because as you add more, you expect stronger response.
Yeah, but when you don't add anything, when it's a so-called DMSO experiment, then that point does not arise, right? I mean, there's no different concentration. It's solvent.
Right, but DMSO itself would have potentially different effect depending on you're using one cell line, so it would be maybe all things that are related to, to just time and protocol and other things that can contribute.
Yeah.
And
okay. No.
If you're using low concentration of your actual perturbations, your DMSO may seem to be, in some instances, almost as big in terms of what it does to the system as the compound, especially if the compound doesn't do much, because that's another thing.
I actually noticed that the DMSO distribution really kind of localized with your 3 different experiment clusters. So I have a feeling that those experiments had different protocols, and that's why the DMSO sort of,
Have different what?
Because they had the clusters with big green, the DMSO was clustering each of them. different concentrations, volumes of DMSO different, was specific for that experiment.
That's very likely might be the case. Yes, that there was actually some plate-to-plate differences that are reflected in this.
Okay, all right. Well, in that case, then the challenge would be to detect the similarity of perturbogens, even though the experiment may have been done on different plates, right? So, okay, very good. No, that's very helpful.
And then say.
By the way, if I could add one more thing, since technically you're in the end you're comparing your results, your clustering results with sort of random clustering. That's certainly valid way. But if you think about these chemical perturbogens providing you with one type of clustering for experiments, right? The same compounds, the same cluster, and then you perform your own clustering based on this projected these vectors of enrichment, right? And you have another type of clustering. And now you have two types of clustering. There's the standard. You probably know that this RAND measure of consistency between two hierarchical clusterings, and the RAND measure can be also this adjusted RAND measure that takes into account these clusterings by chance. And this RAND index has sort of very nice interpretation. If it's close to 1, then you know that all pairs of experiments in both clusterings tend to cluster together.
Yeah. No, I think that's a very valid point. That we should compare these clustering results to that obtained on the raw gene expression vectors themselves, or the signatures that AVI constructs. Each experiment has a signature. You can cluster those signatures, right? You might even already have done that, and so that would be another clustering. And I would be more interested in finding RAND indexes away from 1, which is that there are differences
Right. RAND index provides you certain, at least for me, it's easier to understand what it means to have RAND index of 0.5 versus 0.7 versus 1.
Okay.
So, as opposed to deviation from random number of edges.
Right.
That is expected to be broken.
Okay, I get it. What index? I'll tell. It's RAND.
Okay. Yes, RAND. Yeah.
Guy, I think.
UIUC. Right. Should develop a justice.
Okay. So.
And another question. So if we need to drop the DMSO from this cell line, do you suggest that we also drop all other control experiments in all other cell lines?
What are other control experiments?
I don't know at this.
I think the DMSO was the standard. Yeah.
So here.
Yeah, I would drop the controls. That would be my approach. Unless you use them to construct different signatures, I would drop them from the analysis.
Yeah.
Okay. Thank you.
Okay. So let's skip to point 3 on the agenda, which is more of an update or informational point. So there are 3 people in this room who have been working on understanding the molecular basis of drug response from gene expression data on multiple cell lines. So this is something that started in collaboration with the Mayo Clinic, where they had 300 lymphoblastoid cell lines on which they had measured drug responses of different drugs, and we were trying to find genes that might be correlated with drug response from those data. And this is basal gene expression in those cell lines before treatment. And there was a paper within the last 1 year from Stuart Schreiber's group on this topic. It was in Nature Chemical Biology, basically laying out this very idea that you can use basal gene expression to identify genes that correlate with drug response by having the measurements done on many cell lines. And so the way the three people here differ in their approaches is Casey is working on identifying transcription factors whose regulatory action might mediate drug response, and so he's looking at gene expression data and genotype information, so SNP data on those cell lines, as well as ENCODE peaks to bring the transcription factors in. Sheng is trying to identify pathways associated with drug response, again based on basal gene expression data. And Amin is trying to do gene prioritization, so simply identify a ranked list of genes involved in the drug response. And for all of them, because the molecular basis of drug response is so poorly understood or cataloged in the literature, I mean, the one big problem was, after doing all this, then what? How do you justify what you did was intelligent or useful in any way or correct in any way? And so each of them is trying to see if the genes or pathways or transcription factors coming out of their analysis as being responsible for drug response variation across individuals are the same genes or pathways or transcription factors that come out from the links data as well. Now, the links data, of course, is different in that it's not, it's a control versus perturbation, control versus treatment paradigm, and you see the effects of the treatment. And so you find, if you can try to find what pathways went up or got dysregulated, what transcription factors or what genes got dysregulated on the drug treatment. But that's not the same as asking what genes are responsible for variation in drug response across individuals. So, it's kind of two different biological or statistical questions. But because the underlying mechanisms, they were hoping that there are some underlying mechanisms that are common, both in the response to the treatment and in the variation of the response across individuals. And so they're all using the links data, I think the level 4 data, to basically do various enrichment tests or of pathways or identify the topmost differentially expressed genes in response to the drug treatment in links data. So I just wanted to mention this, that all the discussions we're having are also very helpful in them being able to do these comparisons more correctly. Did I make sense, guys, in what the goal is? Okay.
Yeah. So could I try to recapitulate what you just said? Yes. In my own words? Maybe that would help. So for these leukemia cell lines, you actually have SNP data, so you know the genotypes.
Yeah.
And you have multiple cell lines, and you're then assuming that with these variants that you observe in the different cell lines, you'll have variable expression level.
You have expression data also.
Pardon?
You have gene expression data also.
And you actually have the expression data. You try to explain this variation expression by the observed variants and SNP genotypes.
Right. So you're trying to connect the genotype to the gene expression. That's what EQTLs are, right? And then connecting the gene expression to the phenotype also simultaneously. So you're asking if the genotype is correlated with the expression of a gene, and that gene's expression is correlated with the drug response.
Right.
So that was one of the projects. In the other two projects, we are actually not using genotype data. It's just gene expression data. You can ask, does the basal gene expression across individuals correlate with the drug response across individuals? Does that make sense? This is not even no treatment. You measured the gene expression in those cell lines, but there were 300 of them or 800 of them. and you just measure gene expression, and you ask which gene's expression variation correlates with the phenotype variation.
Mm-hmm. So you're talking about the lymphoma cell lines from
Mayo.
From Mayo, right? 600. I thought it was 3.
There were lymphoblastoid cell lines from Mayo, but there's also a different database called the cancer cell line something, CCLE, which has which has many about close to 800 cell lines on which they measured the basal gene expression.
Yeah.
So it's a different axis of information, right? It's variation across individuals, but not looking at the effects of treatment. And LINX is looking at the effects of treatment, on one cell line or a few replicates of what happened when you treated the cell line with a drug. And so, there's hope that they will share the same mechanism.
Right.
All right. So then the last point is the use of enricher gene sets in knowing. And Charles, might you want to comment on that?
I don't know if you just want to. We were going to possibly process enricher because I believe Avi's done a very nice job of pulling in some data from GEO and putting them into gene sets and making it available. And so we were just going to also incorporate that into the set of annotations that are available in the Knowing Knowledge Network, and use that processing.
So, for instance, that would have already compared the perturbagen effect to the DMSO effect explicitly. No. You said that Avi does this comparison.
So these are not. These, as far as I understand, these do not have gene sets identified by Avi using characteristics direction. These are just ones that Enricher has. These are just the public ones that Enricher has incorporated, and some of the public ones are ones that usually did additional work on making them.
I see. Okay, these are not public.
Well, that would be interesting. At least, like we talked about, internal comparisons to the gene. With the characteristic direction.
Okay.
So that would be that was not data I was planning on processing because I'm not sure where to find it.
All right. So I think those were the points from our side. Some of these I just want to let you know so that you know what we are doing, and and because I expect that you're doing similar things as well, and so I just wanted to keep you informed. Is there anything else you wanted to share?
Oh, what we are doing?
No, I mean anything, any point. I mean, if you want to, if something that's relevant to us, or if there's nothing else, we can stop now. That's fine too.
Well, I think actually, since you started this clustering of Z-score-based signatures, Avi has these other signatures, alternative signatures, derived by a direct comparison with DMSO.
Right.
So maybe it would make sense to actually try to cluster those signatures together, these two types of signatures.
Right. Okay.
And see how the same experiment but two different signatures derived from it.
Okay.
Cluster together or not.
Yeah.
So I mean, there is one fundamental difference, right, between the way you guys are treating these signatures, because what you're treating as a signature at level 4 data is a single observation, like single profile, right? Whereas Avi, in his signatures, makes a kind of level 5 signature, right, by combining the replicates for the same perturbagen drug in his case. And that's something that we have also been working on to a large extent. We are trying to come up with basically an aggregated signature, right, level 5 signature, that would aggregate experimental replicates into a one profile, right? And how do you actually characterize this is a kind of not completely trivial, right? Do you account for variability and all this kind of stuff? How do you account and so on? How do you capture this information?
We'll be very interested in that.
Pardon?
We are very interested in in that level of we don't want to. We also want to work with those aggregated signatures. Because our point was, when you map it from the original gene expression space to the biological process space, the annotation space, then does the clustering reveal other things? And then to us, it makes most sense to work with the level 5 signatures that you're talking about. But we wouldn't know how to make them. So if you make progress on that, we'd love to have that.
So, we made all these signatures using different means of aggregation. Actually, Licia was she's working on knockdown signatures. That was level 6 actually. So in knockdown signatures you have experimental replicates, right, for each shRNA. So you construct shRNA level signature, but then you have multiple shRNAs for the same gene, so you want to aggregate them as well to create gene level signature, right?
Okay. Yeah.
But for chemicals, you stop at this level 5, right? Because you just aggregate experimental replicates.
Okay.
We have these, if you guys want to try to use them,
okay.
And we have them basically created a couple of different ways. So the question is, there are two questions there. One question is, how do you aggregate the replicate? The other question is, what kind of association measure you use for two different signatures, right? And in your case, you kind of first correlate with gene ontologies, right? So that's one strategy, right? About how do you what's your association measure? But we kind of went through a whole bunch of these combinations, of constructing a signature, say, by just averaging replicates, and then comparing or using different correlations as a measure of association, or hypergeometric tests by considering top-down and upregulated genes, then using this connectivity score that Broad is using, then using weighted correlation, right, when you use average signatures, but when you calculate correlations, you weight it by, say, p-value of each gene, right, so that genes that are more differentially expressed contribute more to the correlation measure. Okay. So we have a whole bunch of these tests that we ran, and they're not completely conclusive, but it seems that the best thing for drug signature in our hands was, you simply average the profiles, but you also calculate p-values of each gene against 0, right? So whether the average expression or deviation for each gene is significantly different from 0. And then you have kind of each signature is represented by two vectors, right? One is the average differential expression or average Z-score, and the other one is associated p-value. And then when you associate two different signatures, you use the weighted correlation, right? In which you correlate average expression levels, but weighted by the p-value.
Okay.
And in our hands, this seems to, when you do internal benchmark of this L1000 data, it seems to have worked slightly better than any other combination.
In what way? What was the benchmarking trying to achieve? Because so after you do the similarity, what do you do next?
So the benchmark was basically, so far, was just the internal benchmark. Because what happens with these L1000 data is that the same perturbagen drug, not all of them, but for many of them, they were replicated, say, a year later in a different combination, on a different plate, right, with a different combination of treatments combined on the same plate. So, for example, the way they do it, and it's for Broad Institute, this is the first level of validation of anything new that they find. So, say they find they have a signature they want to compare with this L1000 signatures, right? And so they find top 100 drug candidates. And the first step in that kind of validation process of whether this makes sense or not, they just replicate experiments for these 100, right? Because it's cheap for them. That costs them like, $800. So they just replicate experiments again for these drugs that may be of interest. And then they add these results to the database. So you have these, but when they replicate experiments, right, they again replicate, they perform the whole experiment, right? They create 3 experimental replicates. So you have now drug treatment that was done, initially in one place, in, say, triplicate, and you have the same drug treatment done like a year or 2 later in a different place, right? But again, with experimental replicates. So our benchmark was basically how well you can connect these kind of replicated signature as constructed at 2 different time points in a different experimental context. Does this make sense?
Yeah, absolutely. No, we wouldn't have looked for these meta-level separations among replicates if you had not pointed out. So, there's more information in the metadata that can tell us how to use these replicates for consistency as a benchmark.
So would you guys be interested in, say, some of us here presenting this benchmark and kind of
I was
about how this looks like?
That'd be great.
Okay.
So let's do it next time.
Yeah, so we have an agenda for next time then.
Okay.
All right. All right, guys. Thank you very much.
Thank you. That's very interesting. Really enjoyable having this conversation.
Likewise. Thanks.
All right.
Okay.
Take
care. Before I go, so I'm still working on arrangements with Slavik.
Okay.
If that falls apart, then I'm going to just